DNA replication. The functions of a cell are determined by its DNA. Cells have to reproduce many times. In complex organisms, trillions of copies are made from one original cell. But when cells reproduce, they must replicate or copy their DNA. The structure of DNA reveals how trillions of copies of the DNA in one of your cells can be made and be almost exactly the same each time. DNA replication enzymes. There are dozens of enzymes that facilitate DNA replication, but the key ones that will be discussed are helicase, topoisomerase, DNA polymerase, RNA polymerase, and ligase. College Board requires that you know these enzymes and their functions in DNA replication for the AP exam. First, topoisomerase unwinds the DNA so that two DNA strands can be separated by helicase, breaking the hydrogen bonds between the bases. Topoisomerase and helicase are enzymes. So remember, we have a helix that's twisted like a staircase, right? So first we have to unwind that. That's done by topoisomerase. And then we have to unzip these two strands, and that's done by the enzyme helicase. Once that's done, each strand provides a template for creating a complementary strand of bases. So we have two template strands now that can be used to create a complementary strand. The new strands are made on the inside of each strand. Each new molecule of DNA is made of a template strand, so one of the strands that was unzipped or separated, and a new strand, the complementary strand. The template is used to make the new strand. The template strand is the parent strand since it came from the original DNA molecule. The new strand is the daughter strand, which you see forming here, also known as the coding strand. DNA nucleotide monomers are made ahead of time and stored in the cell. They are attracted to the newly exposed complementary bases in the template DNA strands. The reactions are catalyzed by the enzyme DNA polymerase. The strands are joined together, resulting in the formation of two DNA molecules identical to the original. Many other enzymes are used during the process. Adding new nucleotides. Nucleotides can only be added to the OH end, the 3' end of the template strand. So all new strands are made in the 5' to 3' direction of the daughter strand. The added nucleotides bond with their 5' carbon to the 3' carbon location on the daughter strand. So you can see that happening here. This daughter strand is going in 5' to 3' direction. Semi-conservative replication. The light green arrows here and here show the direction that the bases are being added to each original strand in opposite directions. The result of this process is two new DNA molecules, each having an original parent, indicated by the blue, and a new daughter strand, indicated by the red. This is called semi-conservative because it conserves some of the original DNA in each copy. The molecular process of replication. A DNA strand is replicated in segments. At intervals down the DNA molecule, portions of the two strands separate, creating replication bubbles, which you can see here and here. So it's not the entire strand separated. Either side of the replication bubble, here, 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 and here, is known as a replication fork. The replication fork is where the cell's DNA is unwound in a complex of several proteins and enzymes. The electron micrograph of fruit fly DNA shows several replication bubbles, indicated by these yellow arrows. You can see the bubble here, 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 and here, along with the replication forks, so you can see them at either end, where the DNA is unwound. Eukaryotic DNA has multiple replication forks. Prokaryotic DNA have only one replication fork. We're going to spend a few slides now zooming in on what happens at this replication fork. DNA replication is a precise process that must minimize error. Cells use many enzymes in a complex process to build new DNA molecules from template strands. Topoisomerase, which you see indicated by this yellow bar here, is an enzyme whose purpose is to uncoil the DNA at that point so that each strand can undergo replication. Once unwound, helicase breaks the weak hydrogen bonds that hold nucleotides together, opening up both strands to become templates for new strands. Helicase is the molecule that creates the replication fork. So you see that helicase right here. Single-stranded binding proteins, indicated by these little orange bars, bind to single strands once they're separated to ensure that the nucleotide pairs do not rebind after the helicase passes. Since the strands are anti-parallel, they are arranged in opposite directions. To replicate both strands in the same direction, there are two different strategies, one for each template, a leading and lagging. So the strand on the top is the leading strand template. That's the one that the replication is occurring in the same direction that the helicase is moving. The lagging strand, we can see these have to be put together in fragments. For the template strand oriented in the 3' to 5' direction, so that's this one, DNA polymerase follows behind helicase, reading it from the 3' to 5' direction. Right here, just following behind, synthesizing the new leading strand in the 5' to 3' direction. So our new strand happening continuous synthesis in the 5' to 3' direction. DNA polymerase can only read in the 3' to 5' direction. Or that's to say, in the direction of the leading strand template. The new lagging strand, 5' to 3', has to be made in reverse, starting with RNA primase that adds RNA nucleotides as a primer for DNA polymerase. So you can see here, RNA primase adding these RNA nucleotides as a primer for then the DNA polymerase to use as a template. DNA polymerase latches on to the RNA primers and starts writing fragments of the new strand. As it is going away from the replication fork, it writes a portion, then jumps back in front of it and starts again. As a result, the lagging strand has this discontinuous synthesis of fragments rather than continuous synthesis. The fragments formed by this process are called Okazaki fragments. When the RNA primers fall away from the strand, gaps are left between the fragments that must be repaired. DNA polymerase III adds the complementary nucleotides to the growing strand. DNA ligase finishes the job by joining the single strand breaks between the Okazaki fragments. So you can see the work of the ligase here to bring these two strands together. DNA proofreading and repair. During DNA replication, errors sometimes occur in that the wrong base is added. Most of the time, the DNA polymerase that is adding the bases checks to see that it has added the right base. During DNA synthesis, if the wrong base was added, DNA polymerase will cut it out and add the correct base before the next base is added in a process called proofreading. The error rate during DNA replication is very small. 1 in 100,000 bases are added incorrectly, but are immediately fixed by proofreading and mismatch repair, which we'll describe next. Should the error escape DNA polymerase's proofreading, there are several other processes to fix the DNA. Mismatch repair fixes the incorrectly paired bases right after replication using DNA polymerase again. The next two repair processes occur when DNA is damaged during the cell cycle by chemicals, UV light, and X-rays. Nucleotide excision repair. Enzymes replace the incorrect base by making a cut on both sides of the wrong base and replaced with the right one by DNA polymerase, and then DNA ligase puts it back together. DNA damage repair pathways work throughout the cell, repairing damage due to environmental factors such as X-rays and gamma rays. Polymerase chain reaction, or PCR, is a technique that uses the principles of DNA replication to amplify the amount of DNA available for testing and manipulation. This reaction is carried out by a special machine that utilizes repeating cycles of heat, DNA polymerase, DNA primers, and free nucleotides to build copies of the DNA fragments. This technology enables small amounts of DNA to be turned into large amounts. PCR technology is used for applications such as COVID-19 tests, DNA fingerprinting, medical research, genotyping, and mutation detection. In step 1, DNA is heated to high temperature. The DNA strands denature, separating the double helix. Then the DNA is cooled. Primers and polymerase in the mixture anneal to the DNA, which you can see here. The temperature is increased slightly to increase the rate of replication reaction. So we increase slightly and we get lots and lots of replication. And we just cycle through that over and over and over to get what starts as a couple strands into many replicated strands. The cycle is repeated, doubling the amount of DNA each cycle.